So today I will share some insights on what I've been dedicating my time to the last couple of years. But I will actually start with giving you an overview what has happening in the field. And before I start with that, I will basically share the problem that I encountered with all of you. So, I'm coming from an academic institution where we treat patients with CAR-T cells. And sometimes you want to develop your own CAR, or you have your own target or a novel idea how to make a better CAR. But the problem will always be how to translate this into clinical application within an academic setting, right? Because when you usually transfer that genetic information, your CAR, these 2,500 base pairs into a T cell, usually you use retro or lentivirus. And getting that at GMP grade so you can clinically manufacture, it always costs a lot of money, like $1 million upfront. And also there's like issues when you scale up the production of virus, there's always some batch variability. So you always need to test a new batch. And as I said, the costs are very high. Although, when you're in a company and, you have a lot of money and a lot of people who can do all that testing, we know it's a very efficient way to, in this case, transduce T cells and equip them with the information of a CAR. So yes, if you're in the company setting, and we see this with Axicell, Kymriah, and Lysocell, that this works. But there's also non-viral ways to make CAR-T cells, and these are, in my opinion, highly attractive, especially within an academic setting, or when you don't have that much money and you want to test a lot of different CARs. And one non-viral way is transposition. There's two major transposase, Sleeping Beauty and Piggyback. The scaling is very efficient, and you can get medium efficiency of insertion. But the insertion is random, so you don't know, or it's semi-random, so you don't exactly know where you're going to put your transgene information into. So you don't know which genes will be disrupted potentially by the transposition happening. And there's actually been a report or some issues being reported in one trial, the CARTEL trial from Australia and New Zealand. They used Piggyback for transposition, and they treated 9 patients with lymphoma. And unfortunately, they observed 2 cases of CAR-T cell lymphoma, probably because of their manufacturing process and the random insertion of Piggyback here. So this is actually something that's a no-go. That's something we don't want. And then there's another editing tool, which is CRISPR. CRISPR-Cas9 is the enzyme that I have been using and that I'm referring to now here. And when you use CRISPR, you induce a double-stranded DNA break at a specific location. So you really know where you cut your genome. And of course, you need to make sure that your guide RNA that you use, or your RNP, your ribonucleoprotein, doesn't cut outside the actual on-target site, so that you don't have any off-target cutting and thereby any translocations. You really need to make sure that you have a safe guide RNA that only cuts in the one specific site. But when you have such a guide RNA and a CRISPR-Cas9 RNP, you can actually manipulate the system and then provide the DNA template for insertion using either AV6, which is a adeno-associated virus 6 that doesn't integrate on its own into the genome, but can basically harbor the DNA template. And the DNA template usually has the information of the transgene and so-called homology arms that match the DNA sequence next to the cut site. And the cell is getting tricked. It thinks, hey, this is actually the other chromosome because of the matching sequences, the homology arms. And it will use this DNA template provided by the AAV6 to basically insert your transgene. But AAV6 has some limitations. It's still a virus, so you need to manufacture that at GMP grade, and you will always encounter batch variability, and it's very expensive. Also, there's a limitation in payload size. You can only insert like up to 4.8 kb. And more recently, and I'm going to focus on this part now, there's been some efforts around using naked DNA and also plasmid-based insertion. And these are cheaper and to some extent work well. But there's some struggle, which I'm going to share with you today. Yeah, I'm pretty excited about these platforms because they're potentially useful, especially within an academic setting. So here's what has been happening. In 2018, actually before I joined Stanford, Alex Marson, he has published this landmark study in Nature where they started using naked double-stranded DNA. They would run a PCR, so it's not something you can actually translate into GMP manufacturing, but they would insert using these homology arms here on the left and right-hand side. They would insert this TCR, NY-ESO TCR, into the track locus. And you can see on the flow plots that the T-cell receptor is disrupted, and only the cells that have the NY-ESO TCR, they're positive again for the TCR receptor and the NY-ESO specific TCR. So there's been this other study published by Zhang et al. In Nature, and they conducted a clinical trial actually where they also used double-stranded DNA for the insertion of different CD19. I'm sorry. Jana, ich muss gerade einen Vortrag halten. Okay, kannst du kurz zu Mama gehen? Nein. Okay, dann bring mir das kurz. Ja, dann komm, bring mir das eben. I'm sorry, my daughter. She has interrupted me for a second. So they, here in Zhang et al., they inserted the CD19 CAR into different loci, AVS1, TCR alpha, PD1, B2M, compared them. They also did some in vivo comparison before going into clinical application. And they saw, and this is actually pretty cool, they saw that when you insert the CD19 CAR into the PD1 locus, you actually get better killing in this in vivo model than compared to, for example, lentiviral transduction plus PD1 knockout. But when they went into clinical application, they manufactured for 8 patients. And Jana, dann hol das Handy bitte oder komm her. And they ended up only manufacturing 200 million CAR-T cells within a process that took them 13 days. And for 2 of these patients, they got very low insertion efficiencies, like 5.5 and almost 5% here. And also they didn't meet the target dose. The target dose was 2 million CAR-T cells per kg. And for these 3 patients, 3 out of 8, they were not able to make a dose actually. So there's been some further refinements that have been done by the Marson lab especially. So one was actually that they would add so-called truncated Cas9 target sequences to the double-stranded DNA. So the idea is that when you have your double-stranded DNA, you want to enhance the mobility or the transition from the cytoplasm into the nucleus. And they added these truncated Cas9 target sequences. So the Cas9 can bind to that sequence, but it's truncated in a way that it doesn't cut anymore. So the RNP, the Cas9 plus guide RNA complex, that will only bind to the double-stranded DNA. And then the Cas9 has these nuclear localization sequences that will help the double-stranded DNA along with the RNP to move into the nucleus more efficiently. And then in B, in the upper panel, you will notice that they were able to enhance their knock-in by a factor of 4 to 5. And this is with their NY-ESO TCR. But double-stranded DNA is toxic, as you have seen, especially by the data provided by Zhang et al. You don't make many cells, right? So the Marson lab actually developed this other way to do or to enhance insertion efficiency, but also maintain the cell yield. They started using single-stranded DNA. And then they would add oligos that have the truncated Cas9 target sequences at the end, so the RNP can still bind and enhance the shuttling of the single-stranded DNA. And single-stranded DNA, now you got to believe me, it's always less toxic, way less toxic than double-stranded DNA when you electroporate cells. But there's one problem. If you do this and it works, that's beautiful, but manufacturing single-stranded DNA at yields that are, sufficient to supply large clinical trials or even within standard of care, that's going to be very hard and challenging. So I'm not sure if this is actually something that will advance the field. Although I think this is really amazing work, they were also able to manufacture like a BCMA CAR using a GMP compatible setup. So very high efficiency in terms of knock-in efficiency and cell yield. But again, I'm not sure if you can use single-stranded DNA like for larger applications at some point. And then there's other work, and this is where I also feel, hey, that's very exciting. So this is work from a company called PACT. They recently published, or last year, they published in Nature. They have this, crazy setup, because it's a huge effort where they took biopsies from patients and then checked for neoepitopes and then developed personalized TCRs for these patients. And then they would basically manufacture or generate GMP-grade nanoplasmid DNA. Nanoplasmid DNA has this very short backbone. It's only 400 base pairs. And that nanoplasmid DNA would contain the information of the highly personalized TCR, and they would knock into the TCR alpha locus and at the same time also disrupt the beta chain. And you can tell in C and D here that they actually initially had trouble. So they're sharing here their process versions 2.0, 2.1, 3.0. And initially they were having trouble. 2.0 and 2.1 were conducted on the Lonza electroporator. They were not getting very high knock-in efficiencies and also very low cell yields, as you can see in D. But then they transitioned to another electroporator. I believe it was the thermo xenon device. And they were starting to get higher efficiencies and also higher cell yields. So this is exciting. And they were using nanoplasmid DNA, where you can get tons of DNA, especially if you want to, transition from like a smaller phase 1-2 trial into like phase 3 or standard of care. So this was happening. So this is a little bit of background on what was happening in the field. And I started my postdoc in 2020, and I was thinking another way. I was thinking, hey, we're using homology-directed repair for insertion. That's one way to fix a double-stranded DNA break. But there's another pathway, which is non-homologous end joining. And that one is actually occurring throughout the cell cycle. And it's the predominant pathway, right? Because when you have CRISPR editing without providing a DNA template, what usually happens is not homology-directed repair. It's actually non-homologous end joining. That's why you create knockouts. So I felt encouraged to test non-homologous end joining for the insertion because there's also already been some reports where people would use homology-independent targeted insertion, or HITI, which is based on non-homologous end joining, for the insertion of a transgene into a specific location in the genome of a cell. So what happens is basically you have your guide RNA and your Cas9, you will complex it to form an RNP, and it will cut in the genome. And when you design your template DNA that has the CAR sequence, for instance, you will also have the same sequence that cuts in the genome. You will just add it to the sequence of your plasmid DNA. So the RNP will not only cut in the genome, but also within the plasmid. And what happens is that you will get, in the best scenario, you will get insertion of your transgene into the genome. Of course, there's also the possibility that you don't have any integration, so it's just a knockout. This scenario here, where they show reverse integration, where the spacer from the genome and the PAM from the transgene and the other way around realign and recreate the cut site, this is actually only something that is very hypothetical, because what happens is actually that you have mutations occurring here because of non-homologous end joining, or indels occurring here because of non-homologous end joining, which will disrupt this potentially recreated cut site. So this is what we did, or this is our setup. So we have a clinical trial ongoing. for a GD2 CAR to target a specific type of brain child cancer, DIPG. And we took that GD2 CAR and prepared a few DNA templates, one that inserts via homology-directed repair. It has 2 cut sites, as has been previously shown to be optimal, with homology arms on the right-hand side and the left-hand side. And we were using an exogenous promoter, the EF1 alpha-derived EFS promoter. And then we prepared HITI templates. One has 2 cut sites, so the backbone does not get integrated, and the other one has 1 cut site only, with the backbone of the nanoplasmid DNA getting integrated as well. And the setup was pretty simple. We would take healthy donor PBMCs, enrich them for CD4, CD8 positive T cells, and activate them for 2 days. On day 2, we would conduct electroporation on the MaxCyte device, and then co-incubate RMP and the nanoplasmid DNA for the electroporation to happen. And then on day 10, we basically ran flow. And what we saw was that the insertion efficiencies between the HDR template and the HITI template, in this case, were pretty similar. HITI2 cut site didn't work that much, and this has been reported in the past as well. Suzuki et al., who published the initial landmark study for HITI, this wasn't done in T cells before. They also saw that in HEX cells, HITI2 cut site doesn't work as good as HITI1 cut site. But the most prominent effect was actually that we were getting way more GD2 CAR-T cells when we were using the HITI template. So we were having similar editing efficiencies, but more cells when we used HITI. And that's where we felt, hey, we want to push this a little more, because if we can get the cells, just the knock-in efficiency is not high enough. So we came up with this enrichment strategy, and we called it CRISPR knock-in enrichment, or cement. And for that, we used a system called the DHFR-FS system, or we were basically relying on a mutant version of the dehydrofolate reductase enzyme. And this mutant version of the DHFR enzyme, it causes resistance against methotrexate. And methotrexate is a broadly applied drug. And we used very low doses of that, 50 nanomolar to be precise. And what we did was, actually, in the past, people would have started the enrichment very late after activation, like on day 7 or day 9. And we've thought, okay, if we want to implement this in a, say, 14-day process, how can we optimize the whole scheduling? There were 2 questions. Can we start earlier and then also wash out the methotrexate earlier? And also, can we shorten the duration? So we said, okay, day 7 to day 14, that's kind of established. If we start earlier and keep the duration the same, what's going to be the effect on the cells? And actually what we saw is, when we start on day 3 instead of day 7 and keep the cells in methotrexate for 7 days, we get more robust enrichment of our cells. So we were able to boost the percentage of our CAR-positive T cells from 15% up to 90% or 85% here in these experiments, while the old matrix from day 7 to 14 was having more variability. And then the next question was, can we shorten the whole duration from 7 days down to X days? And what we did was, actually we performed the electroporation, and then we split up the sample into different wells that all had methotrexate, and we would wash out the methotrexate from each single well on a different day, and then run all samples all together on day 14. And what we saw was, by day 4, you get full enrichment, and what happens afterwards, it's not important. It's just 1 or 2% more. So we felt encouraged to actually start on day 3 and then stop on day 7 already. And in the bottom, sharing some flow plots from 2 donors. You can tell it's highly efficient to start earlier and then also stop a little earlier. So we made this more seamlessly integratable into a process. And then again, we felt, hey, this is working well. We can get 80% CAR-positive T cells. The cell yields are fine. Let's scale this up. So what we did next was, we would take a full leukopak and then enrich for our T cells. We would activate 1 billion T cells. And then on day 2, we would perform the electroporation. And you will notice I mean, there's always some contraction by day 2, but there was one donor that contracted a lot prior to the electroporation already, and this is very challenging, but this can happen like in a clinical setup as well, where the donor contracted from 1 billion down to 170 million T cells on day 2. And I will follow up on this donor in a minute. So what we did was on day 2, we would take all these cells that we would get, we would electroporate them as a bulk. And then on the next day, we would actually split up the culture. One half would get methotrexate and the other half would not get any methotrexate. So we could compare the non-enriched versus the enriched ones. And we also had a small scale comparator arm from these donors where we conducted the viral transduction with the clinical grade retrovirus that we have at Stanford. So the insertion efficiencies are shown in the upper part. You can tell that the knock-in efficiencies and the cells had to recover over time are baseline around 15-20%. Post-enrichment, we get up to 80%. And for viral transduced cells, and this is totally fine, we get around 40%. And then when we're looking at the cell yields, depending on donor, donor 3 is the one that contracted a lot. We were still able, after 14 days, to get 500 million CAR-positive T cells. For the other donors, we got 2 and I think almost 3.5 billion CAR T cells. So this is a pretty robust process, especially if you think about what happens after electroporation. We were still able to get healthy cells that recover despite the electroporation and despite being co-electroporated with DNA, which is always toxic. And we tested these cells, compared them to viral transduced. We didn't see any changes in the memory phenotype, nor in the exhaustion marker panels. They were able to secrete cytokines after co-cultivation with NALM6 tumors that have GD2 overexpressed, and with the SY5Y cell line, which is a neuroblastoma cell line. And we also did co-culture assays where we ran IncuCyte. It's not shown here, but those were efficient as well. And then we actually moved into an in vivo model. We took this SY5Y neuroblastoma cell line. We injected the tumors IV. And what happens is they actually give rise to tumors in the liver, in the bone marrow, kind of matching neuroblastoma to some extent, especially with the bone marrow lesions. I felt like it's like a almost orthotopic model. And then 7 days later, we would inject our CAR T cells. And we didn't see any difference between the viral transduced and the knock-in CAR T cells. And then we tested or made sure that we don't have any genotoxicity, and we ran copy number assays, on-target copy number assay where the primer binds within the track locus and the other primer binds within the transgene. So only the knock-in cells will actually show the signal, and we saw that we get mostly a copy number of 1, slightly above sometimes, which indicates mainly monoallelic knock-in and some biallelic knock-in, so that you have CAR in one allele and perhaps in some cells in the second allele. Also, we ran this RAM-seq assay where we basically in silico predicted where does our guide RNA cut, potentially just based on the sequence of the guide RNA. And then we took all these potential off-target cut sites, and we ran this NGS panel that is like a pooled NGS panel. It's called RAM-seq. They have like a proprietary primer where you can pool all your reactions, all your primers, and then run next generation sequencing. And we saw that when we compare our knock-in cells to the mock cells, so the x-axis is unedited. Those are the mock cells, and they're compared to the knockout cells, the enriched knock-in cells, and the non-enriched knock-in cells here. We see that only the only indel occurring specifically in the edited cells is in track. There's no edits occurring in our cells that got the RMP. That is not, it's specific to those. It's not shown here in the slides. We also ran insertion site analysis, and the only location where we inserted into is actually the track locus. So we also made sure that we just inserted into the track locus. It's not going elsewhere. And this is where the study ended, and we've felt that HIDI can actually provide high cell yields, and the cells are happy even post-editing. They're fully functional. Cement CRISPR-Knock-in enrichment is a powerful tool to enrich for your CAR-positive T cells. And HIDI might also provide new avenues to be explored in an activator-free setting where you take naive T cells. And you could actually also generate like fully naive CAR-T cells. So that's kind of where we are at. And I would love to thank a lot of people from our process development team, Vimal, Nikos, my PI, Steve Feldman, Crystal Makel, and also Elena Sotillo for their advice. And my work has been funded by the German Research Foundation, and I'm about to go back and start my own lab. So yeah, thank you so much for your attention.